a plant pathologist who specializes in fungal pathogens as well as oomycete pathogens, and he's going to be presenting on his research titled Screening Phytophthora rubi for Fungicide Resistance.
Good morning. Thank you for having me here today. Today I'm going to be presenting preliminary results on fungicide resistance screening that we were doing for Phytophthora rubi of red raspberry. Closer to me. Better? Okay, it's not. Just give me a signal if it sounds like I'm screaming at you. All right. As with all research, this was a team effort. Most of our funding for this came from the Washington Red Raspberry Commission, and some of it I went a little overboard on obtaining isolates, so I analyzed many more isolates than I was supposed to, and the rest of the funding came from USDA ARS. Chris Benedict, Lisa DeVetter, Tom Walters, and Inga Zasada were big help in finding fields to sample for Phytophthora rubi. And of course, a big thank you to all of the red raspberry growers who participated and allowed us to sample their dying red raspberry plants. I really appreciate it. The only way that we can get isolates is to work with you, and I hope this information is valuable for you. And lastly, I want to thank Brian Beck and Kim Graham, who are in my lab, and they helped process all the samples and run the fungicide sensitivity assays. So my general mode of action when I give a talk is to give a little background into the pathogen, why it's a problem, the typical kind of symptoms it causes, and then I'll talk about the fungicide resistance study, how we conducted it, and what are some of the preliminary results, and then I'll summarize everything at the end. So the background information is that Phytophthora rubi is a soil-borne pathogen. It's related to algae, and because it's related to algae, it loves water. And it's very, very common in the red raspberry industry. It's in many of the fields of those of you who grow red raspberries. We know this from Tom Walters' work with Jessica Gijo, one of his students. We know this from surveys that I've done looking for Phytophthora root rot. It is basically, if you're growing red raspberries, you have a high likelihood of having this pathogen somewhere in your system. It survives for years in the soil. If you take a look in the lower right-hand corner, these are oospores. They have a very thick cell wall. This makes them resistant to environmental degradation. They last for a long time in the soil. It makes them also a little impenetrable to various chemicals. That can make it very difficult to control. Disease tends to be, Phytophthora root rot tends to be very, very severe on heavy wet soils. So if you've got a heavy wet soil with a lot of clay, root rot is going to be worse for you. And this again harkens back to it's related to algae. Algal-like organisms love water, so does Phytophthora. And damage can be so devastating. This is one of the most devastating diseases I've seen in the red raspberry industry. There's nothing that quite does in this lower picture, almost 100% loss here. You can see healthier plants in the background, but it's really bad when you lose most of your plants. So I talked with Steve Midbow and a number of other people who work on red raspberries, and I got an idea of what are the most common fungicides that are used in the industry. It sounds about 30% of you are using mefenoxam, which is the active ingredient of Ridamil, and another 80% of you are using phosphorus acid products, things like Aliet, Agrifos, Rampart, and these are the two most common fungicides used to help control Phytophthora root rot. And fungicides are just one way of controlling Phytophthora root rot. So we looked at these for fungicide resistance, and Jane Stewart and Nick Grunwald's lab down in Corvallis, Oregon, actually did a preliminary study. She looked at only 4 isolates from your industry, one from each county of Whatcom, Cowlitz, Clark, and Skagit. So very, very small number. But she looked at those and she found that all of them were very, very sensitive. This number, 0.062, that is very, very low number. And the higher the number is, the more resistant the isolates are. So as a comparison, the 100 micrograms per mil, that's considered very resistant. She found 0.062, that's way smaller than 1 microgram per mil. sensitivity. So the 4 isolates she tested were very sensitive. Well, it turns out 4 isolates isn't very representative of your industry. There's more than 4 fields out there. So my job was to go out and find a lot more isolates and see if fungicide resistance is out there from a much larger number of isolates. So my methods were to go out and target fields with Phytophthora root rot. I did most of my sampling from March through June. This is when most of the symptoms are there, and it's the easiest time of year to isolate Phytophthora rubi. We tested different fungicide concentrations. What I'm going to be presenting today is just mefenoxam results. Mefenoxam is the active ingredient of Ridamil. It's the most likely fungicide for resistance to develop against because it's only a single mode of action. Phosphorus acid has multiple modes of action. It's a little harder to break and develop a resistance to that, but resistance has developed to phosphorus acid in other systems, so we will be testing that as well. Give you an idea of some of the symptoms that we looked at. This plant, second from the right, is slightly off color. It's yellow. It's what we call chlorotic, but a nicer, easier term is yellowing. This one has Phytophthora root rot. This is one of the things we were looking for in sampling, and that would be a plant I would sample to find a Phytophthora. Another good characteristic is this reddening or browning of the leaves. These are very, very characteristic root rot plants. Another good symptom in June are these half-alive, half-dead plants. You can see about half the leaves on this or more are brown, but there's still some living tissues there in the center picture. And is the pointer showing up using the mouse pad? Yeah, sort of. And then this plant in the right-hand side, that is also still half alive, but it is going down fast. You also see symptoms of scorch where there's just not enough water getting up into the leaves, so the outer parts of that leaf begin dying off. These are very, very classic Phytophthora root rot symptoms. And of course, in the later stages, you start getting those main branches or main canes wilting and dying, and you often see death really, really taking off just as those fruit are starting to be produced, so you can lose a lot of yield there. And finally, where I am isolating from is down near the base. You can see on the root here on the left-hand side, there's this nice lesion after you scrape off the bark. That is the ideal place to isolate Phytophthora. And then on the right is this delicious, juicy red lesion. And you scrape under the bark, you can see a little bit of water oozing out. Very, very characteristic Phytophthora lesion, and the Phytophthora practically leaps out of these into Petri plates. So we went to a number of other counties. We went to as many fields as we could, and we wanted to hit a number of field types. We wanted to make sure we encompass the range of field types that growers are encounter. So we did everything from mostly healthy fields to really, really bad root rot fields. This is an example of a healthy field that we went and sampled. And as you start walking along, eventually you find these gaps in the field. These gaps are very characteristic of Phytophthora root rot, and the plants on either side of that gap are great places for me to isolate Phytophthora root rot. This is another primarily healthy field where on the left-hand side you see one plant beginning to go down, and then there's a beautiful healthy plant in the middle, and then on the right-hand side there's a plant that just died. This is very, very characteristic of the healthy type fields that we sampled. This field shows a disease pocket where the low-lying area where the water collects is where the disease is most severe. So on the right-hand side in the foreground, there's this lush, healthy, beautiful plant. And then as you go toward the left-hand side and start going downhill, you see these plants in the center are sort of brown. And then way in the background, as you start going back uphill where the water drains out quicker, you see healthy, beautiful, lush green plants again. So I was sampling in these disease pockets in low-lying areas of the field. So very, very common on the root rot side of things. Horsetail is often associated with fields that have high root rot pressure. Horsetail loves excess water in the soil. So does Phytophthora. So these fields filled with horsetail are often the fields where we found the most Phytophthora root rot. And those big patches where nothing grows and it appears to be all horsetail. It's because they all died from Phytophthora root rot. Here's two other fields where there's high horsetail pressure. Here they are just coming up in the spring on the left-hand side, and here are the cones as they reproduce on the right-hand side. Most of these fields were heavily decimated by Phytophthora root rot. So if you've got horsetail, you've got conditions that are very conducive to Phytophthora root rot. The other big characteristic that is associated with root rot is a heavy soil with a lot of clay. So when I see soil starting to clot up into big, hard clods, I know that that's a site that root rot's going to be more of a problem. And here, this first row of plants is all dead from Phytophthora root rot. And you can see the broadleaf dock that is filled in the spaces as those plants died out. And thank you for telling me which dock it is. And here is another field, again, a very heavy soil, almost 100% loss here. You see the healthier plants in the background. And here's another shot. Just, the field is just so heavy that it's a great root rot site, bad for red raspberry production. So in all, result sides, we went to 28 fields, and this includes revisiting some that Jane Stewart did with Nick Grunwald back in 2014. We concentrated in Whatcom County. Most of the fields are there, 18 fields there. We've got good representation in Skagit County, where we went to 5 fields. A few down in Clark County. We picked up an extra one in Puyallup, and I wasn't able to get back to the one in Cowlitz. So our goal was 50 Phytophthora isolates. Like I said, I went overboard, and we ended up with 153 isolates. I feel like I got a very representative group of isolates from you guys. And I just want to say, I realize isolates is a bit of jargon. Think of isolates like individuals. So just as we're all individuals of a human species, isolates are all individuals of Phytophthora. So just so we're all on the same page jargon-wise. And our plan is we've just finished testing mefenoxam. The next one we're going to is phosphorus acid, and then we're going to pick up a couple extra fungicides that aren't registered for Phytophthora rubi, but if resistance ever develops in the future, it'd be nice to know what fungicides we might be able to go to for control. So those of you who are at the red raspberry grower meeting earlier in the year, remember that I said we had one very resistant isolate and one moderate isolate. So when you look at this chart, the higher the bump, the more resistant the isolate is. So we had one that was resistant to 200 micrograms per milliliter of mefenoxam. That's very, very high. The good news is that isolate has completely dropped out. I did some reanalysis because I thought this is a very unusual observation, found out it's not Phytophthora rubi, it was misidentified. So that gets to drop completely from the study. We don't need to worry about that one. The one that we are still working on is this moderate one over past the 80 mark, around 50. That's like a moderate resistance. We're still working with that. That isolate is not cooperating with us, and we're retesting it just to see if it is indeed moderately resistant to mefenoxam or not. But if we look at all of the isolates, this was midsummer, we hadn't completed everything. If we look at all 150 plus isolates, what we see is that most of them now, looking at all of them dropping out, those two, that high one that wasn't Phytophthora rubi and then the moderate one, now we see that the highest resistance only goes to 5. That is still very, very low. All 153 of them are mostly sensitive to mefenoxam. So we're not worrying about high resistance at this time. What I want to, and then recall Jane Stewart said the average resistance or toward the higher end. What I want to point out is those in red. Those with the red arrows all came from one field, and we did some sequencing and we found out those are not Phytophthora rubi. They are a completely different Phytophthora species. They're related to Phytophthora megasperma. That's still in taxonomic flux. But there is another Phytophthora out there. Phytophthora megasperma has been reported as a pathogen of red raspberry in the past. But this is the first field that I've seen where most of the isolates coming out were Phytophthora megasperma and not Phytophthora rubi. So we've got one field like that. And then I also want to point out in blue, these were all isolates from one field that are coming out at the higher end of the sensitive spectrum. So they're still sensitive, but they're a little less sensitive than most other isolates, and they're all coming from one field. I don't know why, but that's something to be aware of, that there is one field where there does appear to be just a little less sensitivity out there. So in summary, the big messages I want to get across are that, yes, indeed, most of the Phytophthora out there in red raspberry production is Phytophthora rubi. However, there is at least one field that has Phytophthora megasperma, which is a different Phytophthora species. There's relatively little research on Phytophthora megasperma of red raspberry, so I'm not able to tell you a lot about it. The good news is, across the board, all of these isolates are sensitive to mefenoxam. There's no evidence for resistance at this time. We're retesting that weirdo that came out of one field. And one reason I went overboard on the isolates was this concern about that very resistant isolate, which turned out not to be Phytophthora rubi, and then that moderately resistant one. And I just wanted to make sure that if there was resistance out there, that we would catch it. So far, I'm not seeing that. So at this point, things are looking good. You need to continue rotating between fungicides with different modes of action. Don't just rely on mefenoxam. Don't just rely on phosphorus acid. You really need to rotate through the chemistries to prevent resistance from building up. And as I mentioned, the next thing we're working on is we're going to test a subset of those 150 for phosphorus acid, and then we'll move on to the new fungicide chemistries. So with that, I will open it up for any questions you might have.
Looking at your EC50 values, where would be the resistance level for mefenoxam looking at that?
The resistance level sort of varies depending on which study you look at. Most people, or most of what I'm looking at, they start to call it low to moderate resistance around 20 to 50, and then they start considering it highly resistant at 100 and above. It really depends on the researcher. Given that you're applying mefenoxam at much higher levels than 5 micrograms per mil for the EC50, I'm not too concerned about any of these low values that I'm seeing here. So, yeah, 20 to 50 is the start of the low end of the spectrum for resistance. Yes, Tim.
Jerry, do you recommend at this point to combine these different products at the same time for resistance management, or to do them sequentially if you're going to be using these products?
The question was, do I recommend mixing them or applying sequentially? I'm going to say, I don't know. You need to look at the labels. I don't know, and I'm not aware of whether they can be applied at the same time. That's one thing I don't know. And I would say talk to your fungicide representatives and make sure that you are applying it correctly. That's where I'm going to stop.
The fungicide alien is a foliar application because it's a systemic movement, right? So it can go two directional. So Ridamil probably you are in the soil application basically, and the one you are recommending for rotation is Allied, which can be applied as a foliar application.
Right.
So it's two different way of absorbing into the plant.
Yeah, Ridamil moves up through the xylem, through the water conducting tissues, and goes up. So it's generally applied as a soil. The phosphorus acids can be applied either as soil or as foliar, but that moves.
Yeah.
you had a couple of chemicals there that were not registered that you're going to be testing. Do you have indications that those do work on root rot, or that they might be coming up with labels in the future?
So the question was about the ones that aren't, the fungicides that aren't currently registered for red raspberries, and whether they work for root rot and whether they're going to be registered in the future. I don't know if they're going to be registered in the future, but there is evidence in the literature that both of those work against soil-borne Phytophthora root rot in other crops. For example, like root rot of bell pepper, or root rot of, well, bell pepper is the one I remember right now. But there is evidence that they do work against root rot. Right now, I think the pressure is a little off for me testing those other fungicide chemistries, just because I'm not seeing resistance to mefenoxam, which is the product I was expecting to see the most likelihood of seeing resistant isolates. So at this point, I'm feeling pretty confident that we're not at that stage yet where we need to look at it, but I'm still going to continue those.
So we still have huge amount of symptoms and problems of the disease. So is that related to, well, it's genetics partly, but are there strategies we can use to better use these tools?
So strategies to better improve control. Right now, I'm working on rhododendron and root rot of rhododendron as another project. And what we're seeing is that application method matters. And foliar application isn't always the best for control. And that's a different Phytophthora species in a different system. So there are potential things we could do strategy-wise there that need to be studied. The other thing is fumigation as a major method of control. In my point of view, your fumigation practices are targeted towards nematodes. They are not targeted towards soil-borne pathogens. So one easy thing that I see is you need to change your fumigation practices by increasing, my recommendation is increasing chloropicrin, because you're not addressing soil-borne fungal or oomycete pathogens at all. And so that is one way I see of improving control. So different strategies of applying those fungicides, seeing if they work better, and improving your fumigation. And I heard the bell.
Well, let's just take one more question.
Oh, one more.
Jerry, on the sampling sites, were you able to differentiate at all between sites that had a history of Ridamil usage versus sites that have very little Ridamil usage in their history on that site? Or was there information about Ridamil usage at any of those sites previous?
At most of the sites, it was very difficult to get fungicide usage data, but given that across all 28 sites, we're seeing similar levels of sensitivity, I suspect that the pressure is pretty low across all the fields and all the sites.
Well, thank you, Dr. Weiland. This concludes this session. We have a 15-minute break. Also, if you have received a survey, I believe the survey that we're referring to is the one that Michael Dossett distributed in the